Hi, Mr. Az here. Welcome to my review of polymerase chain reaction, or PCR. So you may or may not have heard of PCR before. I suspect that you probably have. It's a common method used in laboratories for a wide-ranging number of applications. Those being the detection of disease. We know this year how useful it's been in the detection of COVID-19 cases. Uh, it can be used to detect genetic disorders, and it's commonly used in DNA profiling, which I will run an additional session on. And DNA sequencing, again, I will run an additional session on this. So in the examples I'll be using in this presentation, I'll be looking at a eukaryotic cell. I'll be looking at chromosomal DNA in a eukaryotic cell. Now, PCR can be used in prokaryotic cells to detect DNA in prokaryotic cells. It can be used to detect mitochondrial DNA. It can be used to detect RNA in viruses. So whilst I'm using this as an example, it is not the only example we can find for PCR. We're going to start off simply by looking at a monogenic condition, which is a condition caused by a single gene. So this is a sequence of DNA that causes a genetic condition. So we have the section of DNA we're looking at in this example. It's small. It's really, really small. Okay, so DNA is small. We can't see it with a microscope. So how do we go about visualizing this section of DNA? Now, I must state in this example, I've snipped the section of DNA out of the chromosome. Okay, this doesn't happen in PCR. I'll explain that in more detail in a bit. But what do we do? Well, if I start amplifying this DNA, this section of DNA, so I'm making copy after copy after copy, we're going to start seeing this section of DNA. So we're starting to visualize it. Some people call PCRs a bit, or make an analogy to being like a photocopier. It's a bit clumsy. I wouldn't use that in your exams, but it's a nice analogy in that you're making many copies of a section of DNA, a bit like a photocopier. So if we make millions of copies of this section of DNA, we have something we can work at visualizing. Okay, how do we go about doing it? Well, the copying, the making of these many copies, is the process of PCR, polymerase chain reaction. Okay, the next bit is how we go about visualizing it, and that is something called electrophoresis, which I will cover in a different presentation. This is our section of DNA of interest. Now, in my example, in the later slides and in textbooks, you will often see the template DNA, i.e. this bit here, being snipped out of the chromosome. All right. Now that can happen. Okay, you can use restriction enzymes to cut out a section of DNA, but it doesn't have to happen. And in PCR, often the DNA, the template, will be part of a chromosome. So our DNA here is part of a chromosome. I have, for ease of representation, taken that section out of the chromosome. So that's our starting point. I hope that is clear. So we look at the actual structure of the DNA of the nucleotides involved. We've got the bases joined by hydrogen bonds. Okay, these are fairly weak bonds. And we've got the sugar phosphate backbone down here, and these are phosphodiester bonds, which are quite strong bonds. Now, if we break the hydrogen bonds, what we're going to do is we're going to be able to separate the two strands of DNA. How do we go about breaking them? Well, what we do is we increase the temperature. And as we increase the temperature, it will break these hydrogen bonds, the weak hydrogen bonds, but the sugar phosphate backbone remains intact. And here we can see if we increase the temperature, the hydrogen bonds are broken, and the two strands of DNA will separate. You may at this point want to go back and revise and have a look over your notes around DNA structure. So we've got our section of DNA, and we're going to separate the two strands through increasing the temperature. The temperatures I'll cover in a bit. We are then going to have something called primers. Now, primers are engineered to bind to specific regions of DNA, and you know the flanking regions of this section. Okay, so you know the sequence of bases, so you can design primers to bind to those flanking regions. Okay, what the primers do is they allow the DNA polymerase to bind to the section of DNA. Okay, so the strand of DNA will have the DNA polymerase bind to it now because the primer's here, and the DNA polymerase will bind to that. Now we will get DNA replication in the 5', if you remember, 5' to 3' end of this strand of DNA, and we'll get replication in the 5' to 3' direction on this DNA strand. What do we need to do this? Well, the DNA polymerase needs nucleotides, okay, to build. They're the building blocks of the DNA. So the DNA polymerase will need these nucleotides. The nucleotides are found here in the solution here. The DNA polymerase will be using the free nucleotides here to build the complementary strand of DNA up here. So I would look over my notes again for DNA replication if you're struggling here. So the DNA replication, remember, goes from 5' to 3' in this strand, and 5' to the 3' on this strand. And we move along, and the base pair is obviously complementary, and we're building new strands of DNA exactly the same way as you'd have with your DNA replication you've learned previously. DNA polymerase moving along here, replicating the DNA in the leading strand, so before from the 5' to the 3' end. You see the DNA polymerase doing the same on the lagging strand. So we've got two strands of DNA replicated there. And we have two DNA sections. We're going to increase the temperature again, and we're going to separate those two sections of DNA. Pop those up there. So what's in our PCR mix? If you remember, we had DNA polymerase, so there'll be a bucket load of DNA polymerase there in the PCR mix, and we've also got free nucleotides. Okay, so we'll just label those up. We've got DNA polymerase here, and we've got our free nucleotides just here. Okay, so if we just move those over there, what else do we need? Well, we need the primers. If you remember, there are those short sections of DNA that are engineered to bind to our flanking regions. So here are our primers, and again, we'll need a load of these. So if I just make a few more primers, there. Now that is what we need for PCR, and that's called our PCR mix. As I said, there'll be loads and loads of these in our mix. After that round, we have 4 sections of DNA. If we complete another round, we'll have 8 sections of DNA. Complete another 16. Looking at the numbers, you might have noticed that we're increasing exponentially here. So after a very small number of cycles, we're getting large amounts of DNA. And at this point, we have millions and millions and millions of copies of DNA. And we can start seeing that. How we visualize that, as I said, is down to electrophoresis, and I'll go through that in a later presentation. So I'll now go through the specific temperatures and the actual process of PCR. We have our target sequence. I think I mentioned it earlier. We call that the template DNA. So PCR is a cycle. This is why we represent it in this circular diagram here, and it's going around and around the cycle. The first part of the cycle is the denaturation stage. So the DNA is denatured, and to do that, as I said before, we increase the temperature to 90 to 95 degrees Celsius. At this point, the two strands of DNA separate, and we have our template DNA here exposed. The astute among you might have seen a problem here. At 90 to 95 degrees Celsius, we have high temperatures of which normally enzymes cannot work under. Okay, we know that enzymes are proteins. DNA polymerase is no different in that respect. So how do we find an enzyme that works at 90 to 95 degrees Celsius that doesn't denature itself? Well, the answer to this is pretty cool. We use the DNA polymerase from a bacterium found in hot springs. This bacterium can survive in hot springs at high temperatures. Its DNA polymerase has evolved to be able to withstand these high temperatures. And this was the key to PCR. With this enzyme, we can use high temperatures, 90 to 95 degrees centigrade, to denature the DNA, but we don't denature the protein in the enzyme. We call it Taq polymerase. So from here on, I might refer to the DNA polymerase as Taq polymerase. Taq comes from Thermo aquaticus or T. aquaticus, just the Taq from the beginning of that species name. Taq polymerase. So the second bit of the cycle, the PCR cycle, we drop the temperature to 55 to 60 degrees Celsius. And in doing so, we allow hydrogen bonds to form. And those hydrogen bonds will form between the primers and the template DNA. Okay, so we have primer annealing. So we call this the annealing phase. The primers anneal to the template DNA. So on to the third stage now, the final stage in this first cycle, the DNA synthesis phase, where the Taq polymerase will now synthesize new DNA. For this, we increase the temperature now up to 72 degrees Celsius. So to summarize, we have our PCR tube. Into our PCR tube, we will have our template DNA. Now that could be it could be a cell. If you're looking, in forensics, you might be looking at cells for target DNA, and I will explain that when I talk about DNA profiling. It might be a section of DNA that you've cut out of something using restriction enzymes. It might be, if you're having a COVID test, it could just be the saliva that will potentially contain the COVID virus. That's the template DNA. So that goes into the tube. Also into the tube, we'll be placing or will be containing Taq polymerase. There will also be some free nucleotides and also finally our primers. So that is our DNA mix. That goes into the tube, and the tube will then go on to something called a thermocycler. So these are thermocyclers. This is an old photo. This is from my old job. I hope Nilly doesn't mind me using her photo here. But these are a bank of thermocyclers. They're simply blocks that you place your PCR tubes into, and the machine will just raise and drop and raise the temperatures to the required levels for the required numbers of cycles. These are thermocyclers, so it's quite an automated process. On that note, I'm going to leave it there. I would suggest, going through this, that if you haven't already, it'd be a good idea to look back over your notes on DNA replication and DNA structure. And then we can move on to the next stage, which is how we visualize the products of PCR. And for that, we need to look at electrophoresis. Thank you for listening. I just wanted to make a quick addition. Some students have asked how DNA polymerase knows, want a better way of putting it, when to drop off when it gets to the end of the target sequence of DNA. Here we can see our target sequence of DNA in green, and we've got the flanking regions here, so the rest of the chromosome, or whatever is the sequences within, extending beyond that. So what causes DNA to drop off in the end? The simple answer is nothing. Okay, the solution, or I suppose the answer is a mathematical answer, and we'll just investigate this. So we have our template DNA here. We have DNA polymerase binding to the primer site, and then DNA extension will occur. And as I say, nothing is stopping DNA extension. So DNA polymerase will continue and continue and continue until the temperature changes effectively. So I guess polymerase works at about 1,000 nucleotides a minute. So depending on the cycle length, it will just continue, and then the temperature changes. And what we get, so after one cycle, we have what we call variable length DNA fragments. So we can see here we have one variable length DNA fragment and another variable length DNA fragment. So now going into the second cycle, we've got DNA denaturing and separating, and then we're getting polymerase binding again, and we're going to get extension, and extension will happen again along here, DNA polymerase, and as I said, nothing stops it continuing, and we're going to get DNA polymerase here on the lagging strand going in this direction. Of course, it gets to the end. Now this is the point where we're starting to get some actual fragments of target DNA. And on these two strands, we've got DNA polymerase. And in that direction again, we're going to make a copy of the template only DNA, and this one we're going to get the variable length DNA fragment created. So if we ramp this up and move it through the second cycle, again denaturation, and we're going to get a result there. And as we move through third cycle, we're increasing at each of these. You'll notice the number of template DNA versus the variable length DNA fragments. Okay, so if I just highlight here, variable regions are here, 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 and here. And of course, our target DNA regions are there, there. So after 4 cycles, we've got 8 variable length DNA fragments and 8 target length DNA fragments. And as we move through the next cycle, you can see the numbers flipping. So at this point, we're starting to get more target DNA created, and the variable length DNA fragments just remain at quite low levels. So after 5 cycles, we've got 10 variable length DNA fragments and 22 target fragments. And if we ramp up beyond the 6th cycle, you see the numbers of variable length DNA fragments increases by 2, and yet
the target sequence increases at a greater rate. And if you like maths, the numbers are n being the number of cycles. We've got 2n, which will give us the number of variable length fragments at the end of the cycle. And to get the target DNA fragments, we do 2 to the power of n minus your number there, which is 2n. Okay. So if we do an example, so we can extend this to let's say 30 cycles. So after 30 cycles, what will the number of target length DNA fragments be versus the number of variable lengths? So just pause it for a second and give that a go. Okay, so for the target length DNA fragments, we would do 2 to the power of n. So here n is 30. We can see n being 30 here. And that gives us a massive number, which equals 1.1 times 10 to the power of 9, which is an absolute whopper of a number. The variable length fragments would simply be 2n. So 2 times by 30, which is 60. So you can see after 30 cycles, we've got an absolute colossal number of target DNA fragments and a completely almost irrelevant number of variable lengths.